So I think everything seems to be working again. Yes, I can hear myself. So hi, hello, and welcome again. Microbehunter here. I'm Oliver. Welcome to another Saturday Microscopy live stream today. A special request live stream. I have to tell you a little bit why. Special request because yesterday my students, I'm a teacher as many of you already know, asked me, well, what am I going to do in the live stream today? And I said, well, I have absolutely no idea, but why don't you make a suggestion? And because we were just talking about osmosis and diffusion in the class, yeah, somebody suggested, well, can you do some kind of an osmosis live stream? And I said, well, actually, I've already made several osmosis live streams before, but I don't know. Why not try it again, and why not put a few other things with salt under the microscope and see what happens, right? So that's why we're here today, and I've actually made a whole list of organisms: onion, cheek cells, blood, nematode worms, Aeolosoma worms, yeast, spores, and Daphnia. Many organisms that I can put under the microscope and add a little bit of salt solution. Here it is. Yeah, concentrated to diluted salt solution, and then let's see what they're doing, right? And I have to tell you, yeah, they're not gonna like it. Okay, so that's a little bit the idea. Usually the first couple of minutes I always say hello around the world, but today I want to do the following. I want to actually do maybe I want to show it to you first, and then maybe later on after an hour I would like to then read the chat. So I want to separate this a little bit, so please do not be surprised if I don't immediately respond to your questions or comments. I will try to do that later. I will try to keep it a little bit more separate. But at the beginning, usually it's a pretty nice tradition that we say hello around the world. Okay, yeah, even in Esperanto. Yeah, wäre es das Tempo, danke, Oliver. Yes, yeah, thank you very much as well. I do also speak Esperanto, the constructed language, and there's a comment even in Esperanto here. Yeah, hello from Northern Germany, from Vietnam. Okay, from the United States, from Hungary, Budapest. Okay, and yeah, from the UK. Very nice. Yeah, Lebanon, Netherlands. Yeah, Peru, so all around the world. So yeah, what languages? I don't speak most languages. I speak German, English, and Esperanto, and yeah, that's enough for me. I learned a few more in school, but yeah. So what I'm gonna do is the following. I'm just going to dive right into it, and I'm going to just, yeah, even at the risk that I'm showing you things that some of you might have already seen before. Yeah, I'm just going to pretend like it's the first time ever, right? Because some people actually might indeed not have seen the things before. And a few of the samples that I have here, I did not add salt yet, so we're going to see what happens, right? So very briefly, what's important to know is that cells, living things, are, yeah, some of them quite pH sensitive, so acid base, right? That's one thing. And the second thing that living things are sometimes very sensitive is salt concentration. Yeah, so-called the osmotic balance is very important in cells. So if you put a cell into a salt concentration that's too high or too low, some of them don't like that, right? They're either going to explode and blow up and explode, or they're going to shrink. And some cells actually, like I'm going to show you later, the cheek cells seem to be surprisingly salt tolerant. Yeah, but with the onion here, the red onion, that's actually a very nice example. It's actually a very nice example where you can actually see the shrinking of the cells quite nicely. And yeah, so that's something that I just want to show you. And as always, I want to show you also the preparation techniques. So in case some of you want to try this at home, yeah, it's fine. Yeah. So yeah, and as I'm reading right now in the comments here, yeah, usually, of course, even if I repeat the experiments, it's never going to be 100% the same. Yeah. So usually what I do is I try to preserve as much of the onion as possible, because ultimately I do want to not throw it away, right? It is food. So and that's why I try to be as how do you say, as clean as possible, so that I'm able to eat it again. And the red onion is indeed the, how do you say, the subject of choice because of its surprise red pigment. So I'm going to put the onion away here. Yeah. And what we want to do is the following. And I'm going to now make it a little bit bigger here. Okay. Yeah, you see that if you've got those layers here, there's this inside layer. I have to see that's already out of focus again. Yeah, let's make it higher. Does it focus? Yes. You can see, of course, this is inside. Sometimes this inside layer of onion, the onion skin. Sometimes, or maybe it's yeah. You have to look for it. Sometimes it's already went off. Yeah, and yeah, here it is. Yeah, see, there's this thin layer, but that's not what we want. This is transparent layer of of onion cells, which is difficult to get off. Yeah. But what we want is we want to have see this one over here, right? Do you see this? Yeah. This is a transparent layer of cells. You can look at that if you want to. That's fine. That's usually the first thing that you put under the microscope with beginning microscopists. It's a cell layer thick, very easy to observe. But that's not what we want, right? So this onion skin over here is quite nice to look at, but not interesting right now because it's transparent, and we want to look at the red pigmented layer here. The red pigment, it's called anthocyanins. These are also pH sensitive, some water soluble pigments. But the problem that we have here is it's difficult to get it off. I'm not able to get off a layer, so I'm going to show you a little trick. What you do is the following: is you take a knife, you cut into the flesh of the onion. Okay, kind of generously maybe even, like this, right? And then you put your thumb over it and you really press hard, and then you carefully try to rip it off. If it works, try to rip it off. Okay, so that's it. And many of the cells are gonna break, but some cells actually over here are gonna be intact. And this was not a good one because it's trial and error, right? So let's try it again. So I have to, you see over here, there's this transparent layer of cells on here that I want to get rid of. Yeah, okay. And over here, again, I'm going to go into the flesh of the onion, and I'm going to carefully try to rip it off. And this one actually looks much better. Okay, you see there, the white spots that you see in here, these are the cells that are broken open. I'm not so interested in those, and the rest, many cells are still intact. And what I have to do now is the following. I have to put it on a microscope slide and have to cut away the thick part of the onion. Just going to use, which one? Yeah, this one over here. Just going to put it on here. Let's focus again. Here we go. You take a scissor. Up, I dropped it. Okay, and we're just going to cut away the thick part here. And we're going to try to put it on here as flat as possible. Okay. Okay, so that's it. Yeah. You see that I'm not adding any water or anything. Yeah. So this is more or less as flat as possible. And you need a little bit of patience. I don't want to touch it too much. I do not want to damage it, right? Yeah. So that is number one. And number two, all I do now is the following: is I will add a cover glass directly. Okay. I know normally you would add some water, right? I'm not going to do that. I'm going to add directly a cover glass. Press flat a little bit, and we're going to put it now under the microscope, and we're going to check if it actually, if you're able to see a couple of cells. And then I'm going to be adding salt water, and we're going to see what happens. Okay, so that's kind of the idea. Yeah. That is the thing. Occasionally, I do read a little bit the comments here, but again, I do want to maybe respond to them later on. And if you have any questions or any comments, please always write at Microbehunter. Then it's easier for me to actually see if a comment is directed for me, because sometimes in the community people also want to chat with each other, right? So I'm going to go over now to the microscope view. I'm going to remove the DIC. So I'm just going to use a regular bright field. I already have the 10 times in here, and I already see that it looks quite okay. Right? I need to focus a little bit. Yeah. So again, this is without having added any water. You see that over here, those white cells that you see, these are basically damaged. So they kind of the pigment ran out, right? So you see the nice cell wall, really, and there are many cells that are still red and intact, and that's what we want. And what I'm going to do now is here there are a lot of them here. Right, I'm gonna go up a little bit with the magnification. So this is now kind of how do you say, a regular bright field. I mean, if you do look carefully, then occasionally you are able to also see a nucleus. Just for those of you who wanna know. And yeah, here nucleus, nucleus, nucleus. Yeah. And I do actually wonder if some of my students actually joined in, because some of them said that they actually want to join in into the live stream. I told them it's going to be quite late at night where I am, but they said, it's going to be fine. So let's see how many actually showed up. Yeah, so, and that's basically, lots of air bubbles, of course. Yeah. So, and what I'm going to do now is the following. I'm going to add now quite a concentrated salt solution. I mean, I've got different concentrations here. I've got it from concentrated, and I always reduced the concentration by half. So it goes from 100% to 12.5%. But you know what? I've always used 100% because the effect is very quick. Yeah. So I'm just show you what I'm doing. That's it. Yeah, so I take a little bit of here. And now I'm going to add it, check up here, to the microscope slide, and then the capillary action is going to draw it in. Yeah, So let's have a look, and of course I have to be careful that I'm not touching the objective here. And then it's going to go quickly out of focus as well, and I have to probably refocus everything. Sometimes we have to see if the capillary action takes. Yeah, let's see. Is it being drawn in now? But what's going on here? Up. Okay, I think now the liquid. You can actually see. And then let's see. Let's hope that this actually works. Okay. Let's hope that this actually works. I might go up with the magnification a little bit. Yep, and I can already see stuff happening. Okay, you might want to look a little bit. Let's go up with the magnification. Here we go. I might have to add a little bit more, but you can actually see how, and then I will explain a little bit what happens. Maybe honestly, I've seen this a little bit stronger already in some other cells, but you can actually see that there are these white areas now, and that the central part of the onion here, the red part starts to pull together. You see those things happening, appearing, right? So the inside starts of the cell over here. You see it quite nicely. Do you see how the inside over here starts to shrink? Yeah. This is called plasmolysis. Oop, I bumped it now again, right? Here. This has here you see it quite nicely. Here on the side. That's where the here it has already progressed quite a bit. Yeah. You see it over here. The central part has actually shrunk now. Right, and over here it's bright, and on the outside here you still have the cell wall. Yeah, this is called plasmolysis. Yeah, let's go down with the magnification so we get a better overview here. Here, that's it. Yeah, yeah. So here it didn't quite reach, the salt didn't quite reach it yet, but here you can actually see it already that it started to shrink quite a bit. Yeah, and the important thing, or kind of this educational aspect here, is actually to show that the cell wall is strong and stays, so the actual size of the cell doesn't change, but it's actually the inside of the cell that starts to pull together. And another thing that you might realize is the following, and that's also something, is that the inside of the cell, maybe you noticed it's actually become darker, so that the pigment inside the cell here is now actually dark red. Before it was brighter because it's more concentrated, of course. Yeah. So the water was kind of pulled out by osmosis. Yeah. So the cells are now suffering. That's correct. Yeah. So this is so that is the how do you say the thing that happens, and what you have to look specifically, and that is something that we also see over here. And I'm going to go really up with the magnification now, because this is called the plasmodesmata, you can see. And that's also very nicely visible. That's actually, if you look carefully, you're going to see that over here at certain places. So what you have over here, that's the cell membrane on the outside, right? And at certain places, the cell membrane is connected to the cell wall, right? Over here as well, these are like very thin strings. And this is basically where one cell is connected to the other cell. Yeah, so the cells, actually, the cytoplasms of the cell, the inside, are actually connected with each other because they're kind of stretching through and then going to the other cell. Yeah, so there are places here where it does not separate. Yeah. Yeah. So is it specifically the membrane that's shrinking? Strictly speaking, the membrane is separating. The membrane is so thin that you don't see it, right? So, but it's on here. The membrane is separating from the cell wall, right? But at these places here where you see those fine strings, this is where it's still connected to the cell wall and to the next cell. Here you see it quite well as well. Yeah. Here, you see all of those tiny strings here, plasma strings that are where it's connected. Yeah, so that's, and now you also see very nicely how dark the central part has become because the concentration, has gone up. Yeah, so I have to check because I have still so many things that I want to show you. And what I want to do now is the following. I'm going to now try to reverse the whole thing. Okay, by removing the salt and by adding some regular tap water, and then so let's take this away. Okay, so let's go up here. You see the red pigment, and what I have to do is I cannot simply add tap water to this because there's still the salt water around. So I'm going to do it like this. I'm going to do it the easy way. I'm just going to wipe the salt away. Okay, and here I'm also carefully going to try to remove, without actually damaging this too much. Yeah, because I really want to make sure that, So, and we put it again on here. And let's put it in again. And where is the scope? Yeah, here we go. That's now you see it quite nicely, right? And now let's try maybe a slightly larger magnification here. Okay, that's fine. You see, it doesn't look quite as sharp anymore and as clear anymore. And the reason is because I don't have any water. Yeah, but I mean, you get the idea. Again, you see that bumped it. You see again that the outside of the cell wall, the cell size did not change, but the inside shrunk. And now I need some regular tap water. Okay, here I'll just take this, some regular tap water. I need to get also a new tip from a pipette because this one contains salt. And then I'm gonna move on and I'm going to, and then I hope this works. Okay, I'm adding now again. Look at the top. What I'm doing, I'm adding now again some up. And now I add it. Look how it's swelling. It's starting to swell. Look, they're starting to fill out up again. And they start to become round. How interesting, however, is that essentially they don't fill up completely. And occasionally, the interesting thing is they actually pop on the inside. So maybe we're able to see this as well. I'm going to add a little bit more water maybe. Yeah, but you see how they started to swell up again. Yeah. And let's see how this works. Well, I'm gonna go down with the magnification a little bit. Yeah. You see how basically they started to swell up again, and maybe here. Yeah. Yep. There was one that's wearing sunglasses. Yeah. Yes, and occasionally you're able to see the following. Look at those here, right? Sometimes they start to fill out, the whole cell starts to become pink. And that is because the cell membrane ruptured and popped, and the inside started to flow out, and then the whole cell starts to fill up. Yeah. So occasionally you're also able to see that. Here especially. Okay, so maybe we're able to see some cells popping over here. Okay. Yeah. Occasionally you're able, to see that for whatever reason. The membrane starts to pop, and then it starts the content starts to fill the inside of the cell. Yeah. Yeah, but I think you get the idea that by adding again water, you're able to reverse the whole process. Yeah. Kind of fascinating to see these processes of life. Yeah. Yeah, that's a good question. What you're saying, I wonder why they usually don't return to the original size and pop. And that's a good one. I don't know. I've been trying to figure this out as well. Yeah, but I'm a couple of years ago, check the main channel, the Microbehunter channel. I actually did make some very nice videos actually showing the popping process. Yeah, so it's yeah. It was a couple of years ago in the not in this channel, but I've got the main Microbehunter channel where I'm actually made some really yeah large yeah videos of this, yeah, where the cell's nuclei not visible anymore, but yeah, basically the nuclei were visible before, and I think they're just not visible right now because of the lack of contrast, yeah. But maybe you were able to see it a little bit on the inside. Maybe that some of these bright areas that could be the nuclei, yeah. Not sure now, yeah. So it could be maybe the stress from the rapid expansion, yeah. Yeah. But the idea is, yeah, so it started to stabilize now, but the idea is that it is possible to reverse the whole process. Yeah. Let's move on a little bit because I could continue to talk, but I still have a few other things on my list here because some people also wanted to see my blood under the microscope and see how the red blood cells change. But before I do that, I wanted to show you my cheek cells, and interestingly, the cheek cells, when you add salt water, do not change so much in shape, which I considered initially interesting, but then again, it's not surprising because, I mean, we're eating salty food and we're drinking also water, so the change in salt concentration in our mouth is big, right? Depending on what food that we eat, right? So it should not surprise us that the cheek cells inside are very resistant, so to say, yeah? So that is the thing, yeah. So let's try that. I just want to show you. I'm just gonna show you how this works, yeah. So yeah, yes, I'm gonna prick myself a little bit, yeah, with later. Okay, so let's clean up this here, and I'm going to show you quickly again. Again, many of you probably already know that. Doesn't matter. I'm going to now show you how I prepare my cheek cells. And again, this is an example where there is not, interestingly, there was not a big change in how do you call this, in shape, right? So here it is, right? Just a regular for the ears. You collect some cells. So here we go. I am reading. I am reading the comments. Okay, so let's. I mean, there's a lot of liquid already here. Lots of liquid. I'm gonna throw it away. Let's add a. Where's the cover glass? I have to admit, I just washed the cover glasses. They're not perfectly clean, even though I washed them. So I'm going to simply do a little bit of a dry cleaning here as well. So let's add the cover glass. No salt water yet. Let's have another look. And I'm going to now show you phase contrast, okay? Because that's actually quite nice. And scope. So that is the regular, yeah. In that sense, we can already see the cells, but not very well. So you're just gonna, in a second or so, you're just gonna realize how important contrasting techniques are. I can already see them. I wonder if you're also able to see them. Do you see those very these things here? Yeah, these are the cells. Those big things here, these are of course bubbles, air bubbles. But those fine things here, almost impossible to see. These are the cells of my cheek. I did not stain them. So I'm going to show you now how this looks like in phase contrast. Okay. I need to open this. Where is this? Here we go. See? That's the whole point of phase contrast. Yeah. Let me see. Yeah. So you see very flat cells over here, a nucleus, right? Yeah. And I'm going to add now again the salt water. And when I tried it before the start of the live stream, I realized, oops, strange. They don't change their shape very much. Yeah. So let's see if this is still the case. Okay. And I think the reason, I mean, I did a little bit of research and I found one paper that actually said, actually it should change their shape, right? Kind of strange. So we're going to see how this works. Yeah. So I'm just reading here. I need to buy phase contrast unit for my cheapo microscope. Unfortunately, phase contrast is not cheap, and I would probably do the following. You can actually stain them. It's cheaper. You're cheaper off if you actually stain it. Yeah. Let's make it a little bit brighter. Yeah. So, and you're going to notice also another thing, because right now, yeah, you're gonna see, right? Again, check what I'm doing here. Let me adjust the thing here a little bit at the top. Yeah. So, and let's add again a little bit of salt water. This is now again the concentrated one, and I have to see if it's actually able to be soaked up into the. Does it soak up into the? Ah, yes, it did. I have to wait now until the salt solution reaches the cells, but then I discovered actually, not a lot happened. Yeah. But you can actually see nicely the flow. Yeah. And again, I think the reason is because I don't know how the cells do that, but they seem to be indeed more stable to changes in salt concentration, which kind of makes sense, as I already said before, because we're eating salty food and then we're drinking water again, so that the changes in salt concentration in the mouth are big, right? So it kind of makes sense that the cells are able to withstand those changes. Yeah. So yeah. Could the saliva protect them? That's the question. Could be, right? But before I actually this live stream, I actually had a pretty dry sample, so without a lot of saliva. Yeah, yeah. So yeah, just wanted to show this to you that not all cells respond, yeah, equally, and that's interesting. Because, as a matter of fact, in this one publication that I found, they actually said that it does shrink, but mostly not but especially in a substance called polyethylene glycol, PEG. Yeah, and yeah, maybe not so much in salt. Yeah, but I just wanted to show this to you. I kind of found this interesting. Yeah, and now, yeah, you can also see that not evidently some cells have some kind of a way of kind of protecting them. Yeah. Themselves, because it's very different with the red blood cells, by the way, right? Which I'm gonna show you now, because the red blood cells really change their shape. Huh? You won't be able to recognize them anymore as red blood cells. That's dark field. Yeah, yeah, and here again, this is yeah, we're not able to see them here. Yeah, so that's again, those dark spots that you see are the bubbles, and the cells in here. Yeah, very difficult to see. Yeah, that's why I'm showing you the whole thing with phase contrast. Yeah. Here we go again. Where is it? Okay. So that is yeah, the thing. And now I'm always checking a little bit the time, because I've been already talking half an hour. We're just on sample number 2. So let's do some more. Let's have a look at the blood. Okay. This is actually the scary one. And yeah, if you don't like to see blood, then I just ask you, maybe you can close your eyes. Okay. But yeah, in some weeks ago or months ago, I actually did a whole session on blood microscopy. And I don't know how many, I pricked myself, I don't know how many times during this live stream. So it's all a question of practice. Seriously. At the beginning, it really takes a lot of, how do you say, yeah, it's difficult to overcome this instinctive, how do you say, this instinctive resistance of pricking yourself. But it's a little bit like, I mean, I've got contact lenses, right? And putting the contact lenses in, when I got them first, was very difficult. But then if you keep on doing that, then you gain practice. So it's the same thing. You learn to overcome your initial resistance to it. So the first thing that I'm going to do is the following. Look what I bought here. I bought this. When did I buy this? I don't even know when I bought this. These are some, I think you call them lancets. Yeah, and they are used to draw blood. They look like this. They're disposable. But what I always do, generally as a general, this spot here, that's just from the onion. Generally what I do is I always disinfect myself first, simply for it's alcohol. Okay, just regular, right? Simply for the sake of keeping everything a little bit clean. And the wound is so small that actually it will stop bleeding right away again, right? And yeah, so it's kind of not such a big issue. So I have this here, and I need cover glasses. No, I'm going to use this here as a. Not, I'm not going to use those. By the way, in case you're wondering what this blue part is, this is on the glass, and you can actually use a pencil or a pen to directly write on this here, just in case you're wondering. Yeah. So this is a writing surface. Yeah. So but I need a completely how do you say? Yeah. You know what I'm going to do? I'm just also going to wipe this clean a little bit, because I also washed them, so it will be fine. I need another one, another slide here. And how I'm gonna do this now? I need a cover glass. Which cover glass should I use? If you want to get a very thin layer of blood, you have to use large cover glasses. Look at those here. These are huge ones. Yeah? Because if you use a large cover glass, then yeah, the blood will spread into a very thin layer, and then we're able to see the individual red blood cells better. So what we're going to be doing is we're just going to have a look at the red blood cells first. And the important thing here is, well, there are two different ways on how I can do it. I can put a drop of blood here and I can make a blood smear. I think I'm not going to do that. I'm just going to use it directly on the cover glass. So this is now the fun part, okay? And let's do it like this. This is where the psychology now plays a role. This is the lancet. You have to open it. Okay, again, if you have a problem seeing blood, then you gotta close your eyes now, right? Yeah, so this is a needle. Yeah, so this is not only for protection, but also for keeping it clean. Then you do the following. Yeah, there are lots of blood vessels on here, and kind of pushing it in does take a lot of psychology, right? And you don't want to do that. So what I usually do is the following: I go 3, 2, 1, zack, and I just flick myself here, yeah, and it just hurts a little bit, and then that's enough, hopefully, and then you press and squeeze, right? You put a drop of blood directly on the slide here, yeah, maybe a little bit more. This is not a lot. Here we go. Okay, I'm going to simply wipe the rest away. And a cover glass goes on here, look, like this. Allow it to spread. And you carefully lower it. And you can see that capillary action starts to draw the blood across. And it's very thin, a very thin layer. So we're going to have a look at this, okay? Yeah, see? Yeah, it's almost stopped bleeding already. Yeah, so let's have a look at this. Yeah, and then we're going to be adding again a little bit of salt water. Okay, so let's have a look under here, and scope view. Oh, that's dark field still, right? I'm gonna go like this. Ooh, it's too thick. The layer of blood is way too thick. I used too much blood. Let's use bright field. Way too much blood. Not good. I might have to do it again. Because I hope that actually it will start to separate out until you are able to see the individual red blood cells. Yeah, you are now the dots that you see. These are the red blood cells. Okay, good. Some fiber from cleaning it, but I think we now have approximately a single layer. Let's go up with the magnification here. That's already better. Okay. Yeah, let's see if where is it? Here it is. It's flowing. Way too much blood. I don't like it. Yeah, and nearly everything. I think I have to do this again. But at least, I mean, it does look kind of nice if you can actually see the blood flowing. Yeah, it's kind of spreading by capillary action. Yeah, too much. My mistake, my bad. Yeah. In the meantime, you know what I'm gonna do? Well, while you watch this, I'm gonna try to squeeze out a little bit more blood. No, it already stopped bleeding. It stopped bleeding already. Okay, I'm gonna spare you this. I gotta prick myself again. Yeah. So, yep. So I pricked myself again. Now with less blood. Okay. I don't have the stain, unfortunately. Okay, and the point is I want to show you now what happens when you add salt to this. So look, I show you quickly the desk view again. Yeah, so that's basically a much less blood, okay? And let's have a look at this here, okay? So again, the scope view. So I'm gonna change the slide. This was too much. Okay. Okay. Oh, it's still. The blood is pretty thick. It doesn't want to spread anymore. It's kind of interesting. It's even worse than the other one, I just realized. Okay. Here we go. I'm just looking for a very thin. It's too bright. Yeah, this is getting better. You can see the individual cells better. I don't know. Nah. Otherwise, if this doesn't, then I will try the following. Where is this? I'm going to try to squeeze this and maybe spread it this way a little bit. Okay, I'm going to do the following 60 times. Yeah, not very, how do you say? Maybe, I don't know. And honestly, I've seen already better ones. Ah, here, that's better. Look, here we are. Okay. So, and what can we see? Very nice. Blood that starts to coagulate will sometimes stack up like this. This is called a rouleau formation. This is completely normal. Every blood will do that. Why do I have to say this? Because some folks say, oh my gosh, if this happens, if the red blood cells do that, then you've got some kind of weird diseases. No, it's not. This is just a normal, how do you say, phenomenon that happens. So these are basically the red blood cells here, right? Yeah, it's 60 times. You can actually see that at the top, right? And I'm not going to go up to 100 times oil immersion. Yeah. So 60 times, and you have to understand the following, because I've got a very large sensor in my camera, it actually looks smaller than when I look through the microscope. Yeah. So you know what? Why not try also some DIC to add a little bit of color here, because color is nice. Okay, you see, you get the idea. Here we go. You can play around a little bit as well. Yeah. You get the idea. Yeah. So, and now the fun parts, you know what? Why not stay with the bright field, because it's the natural. Yeah. So let's go close the condenser a little bit. So now I'm going to add a little bit of salt, okay? And let's hope that this actually works. Let's see, what is the time? I always need to check the time. Time passes way too quickly. Yeah, and let's hope that this works. I'm going to add some salt now to the other edge of the cover glass, and I hope that it's able to reach the center. Yeah, something seems to be happening. I'm going to go down with the magnification to get a better overview. Here, that's the salt. That's the salt. See, that's the salt. Liquid water spreading. Good. Is that a little more? Okay. Where is it? Here. Let's see until when maybe it's gonna touch the blood soon. I don't know. Where's the blood? Let's go down with the magnification yet further. Here we go. See how it's spreading? Should it be reaching the blood soon? I don't know. Ah, here it is. Okay. And the red blood cells are being flushed away by the salt water. Okay, let's wait a few minutes until the whole thing starts to stabilize. Look at this. Looks pretty cool. Yeah. And then let's have when everything's stable, let's zoom in a little bit more and let's see if the shape of the red blood cells has changed. It should have. Okay. But we might have to, yeah, look, and it's because a little bit diluted, that's good, then they're a little bit more separate. So let's go up with the magnification again. And 60 times. And we're able to see something quite nice. Okay. Very good. This is actually just the way I wanted to show it to you. Do you see that they look kind of weird? Yeah, they don't look like those round donuts anymore, but actually they look like, I don't know, like they have spikes, like on them, right? Why is that? Well, the reason why they look so spiky is because of the salt water, the red blood cells, they lost water and it started to shrink, and now the cell membrane of the red blood cells is too large, and that's why it makes those folds. Yeah, and that's basically, yeah, a completely different shape. That's why if you look carefully, yeah, not only do they look a little bit smaller, but they have these, yeah, they look very wrinkly because they are. Yeah. Okay, this is the effect of salt water on those red blood cells here. Okay. Yeah, and I'm just for your info, feel free to post comments or questions. I'm always reading them, but I'm not responding yet. It's just to keep this first part a little bit, how do you say, compact. Yeah. So this is basically what happens when you mix salt water with blood, and so you see that the onion cells, they respond quite strongly, the blood cells here, but the cheek cells, for whatever reason, not so much. Yeah. Yeah, so that's the thing. Yeah. So I think it's quite quite nice. Of course, I'm not going to do that now because it's really difficult to see if you now add, for example, distilled water to the red blood cells. What's going to happen is they're going to swell up and they're going to pop open. But the problem is you can barely see that, because then when they pop open, you get so-called, they call them ghosts. The red blood cells ghosts. They're basically, all of the content is spilled out, but then you cannot see anymore where the red blood cell is. So it's very difficult and low in contrast. Yeah. So making them shrink with salt water is actually much better. Yeah. So yeah. So I had to prick myself twice, and now I stopped bleeding again, and I think yeah. This was I did a video where I bumped my head getting out of the car. This was many years ago. I did bump my head once and was actually bleeding here. And then I went back and just said, I'm just going to use this now to put it under the microscope, and I made a video. Yeah. Can we add more magnification? Honestly, do I have to? You know what? Yeah, I'm gonna do that, okay? So yes, I can add more magnification. I don't like doing that. I will tell you why. Because I have to add immersion oil, and that's always a little bit risky. But I will do that because, why not? Because, I mean, I need to get some lens tissue paper also ready. So what I'm gonna show you is the following. I'm gonna go now from 60 times all the way up to 100 times, okay? So it's gonna be 40 times more. A percent more. First of all, I need to do the following. I need to remember the position. So I have a focus lock lever. So I'm going to lock the focus. Okay. And then I cannot go back to a lower magnification. Okay. I've got some immersion oil here. And look what I'm doing. I'm rotating the oil immersion objective in place. Yeah. It's kind of very blurry. Yeah, and you see that there is not a lot to see. See, here we go. Yeah. So that is now basically without immersion oil. Okay. So that is without immersion oil. So it looks okay, I guess. But I'm just going to show you how much better it will look with immersion oil. You only do that with an oil immersion objective. Okay. You have no idea what I've already experienced. So folks think they have to add immersion oil to the other objectives as well, and then to make a better picture. You end up destroying the objective. I'm not kidding. right? Yeah, so I'm going to do the following. I remember the position. I'm going to lower, look. Yeah, I'm lowering the slide. I add a drop of immersion oil on the slide, on the cover glass. I raise it up again. I can do that because it remembered the position. I rotate it back and forth a little bit to spread the oil, and I refocus. Yeah, so, and I think you can see it looks way brighter and also more crisp and clear, right, of higher resolution. Yeah. So yeah, and this is basically so we've also done a little bit of oil immersion microscopy as well. Yeah? So that's now the highest magnification that I can get. Yeah. Yeah, and now you can actually see that the red blood cells are completely deformed. Yeah. Yeah. Yeah, I'm occasionally reading a little bit. I'm reading a little bit. Look, I'm focusing now. Do you actually see the cells moving back and forth? Right. That's because as I'm focusing, the objective over the oil applies a little bit of pressure on the cover glass. Yeah. Yeah. So yeah. So that's why as I'm focusing back and forth, it's kind of shaking. Yeah. So I'm gonna move on. Okay. Because I do want to keep a little bit to the time frame as well. And this means that I have to now do the following. I have to clean away the immersion oil. I need some lens paper. Careful. Where's the lens? Here it is. And I'm going to lower it again, the slide, because if I now use another objective, I'm gonna contaminate the other objectives. I don't want that, right? So I'm going to put this away. And I have to carefully wipe away the oil from the oil immersion objective. So you can actually see that there is some oil on here. So that's it. Throw it away. And we're ready to go to the next one. What do I have on here? Yeah, you probably want to. Okay, onion cheek blood nematodes. I have not tried this yet, people. Honestly. So the next one is going to be a little bit of a. Let's put this away. This looks too scary. Otherwise, I'm going to hurt myself. So I'm going to throw it away. So the next one are going to be some of my worms. I need to also put this away here. Just a second. Okay. Got a trash can down here. Worms. Where are my worms? Vinegar eels. You've seen them before. One of my favorite ones. Every now and then I start to pull them out and to show them to you if I don't know what to show you. And I've actually sub-cultivated them. I need to find a jar with worms. Here are my worms. Here they are. They are vinegar eels. They like to have low pH. I have never tested them honestly with salt. Okay, so I don't know. I really don't know how they're going to respond, how they're going to react. I have absolutely no idea. That's why we're doing this today, right? So let's see if I'm able to, I don't know, get some of them out here. Let's move this up. Let's see. Yeah, not too much of this. I don't know. Yeah, maybe. Yeah, maybe. You know what I'm gonna do? Let's throw this away. And up. And I'm just gonna put it carefully under the microscope without a cover glass. Okay. So simply to see if anything is there at all. So let's see if the worms are moving around happily or not. You cannot see anything because it's too bright. Let's turn it down. It's out of focus. Of course, it's out of focus. Okay. Let's go down with the magnification a bit. I do see something moving. Let's get it into focus. Here they are. Okay, yeah, there. I got plenty. Okay. I got plenty of worms. Here they are. Yeah, why do I have them here? They can be bred as fish food. Yeah, so I got them from a colleague of mine at work, another teacher who's got an aquarium, and he's breeding them, and I've been breeding them for 2 or 3 years. Yeah, I've been always sub-cultivating them. Okay, so I always return to those because they're kind of fun to look at, and they like to live in vinegar, so they're pretty acid tolerant. I don't know if they're how or to what extent they are tolerant to salt. Here, that's better, to salt water. Yeah. So that's they're called nematode worms, also known as vinegar eels. Vinegar eels. That's the common name. Tubatrix, I think, is the genus name, and they're very common. They can be bought as fish food, and people who have an aquarium they make basically they breed them for fish food. Yeah. So sometimes they can also be found as contaminants in when you make your own. I don't know, vinegar or kombucha and so on. Sometimes they can also be found there. Yeah. So that is a little bit the thing. And what I'm going to be doing now is I'm going to be adding some salt again. Because why not? Okay. And then let's see if they also start shrinking. And honestly, I'm really honest with you, I have not tried this. Okay. So I don't know if and to what extent or if at all we are able to see a response here. Okay, so you probably don't want to use too much of the salt solution. And I did not add a cover glass now, okay, and I'm at 4 times of magnification. So this means that, yeah, I can just add it on top of them and we're going to see. So a small drop. And let's see how salt tolerant they are. With their wiggling, they are mixing the salt solution anyway. And so far they're fine. Yeah, maybe not all parts reached the salt. Yeah. If not, we're going to add a little bit more, and we're going to see if yeah. I know that, I mean, they are pretty pH tolerant, so maybe they have some kind of a mechanism to yeah, stabilize themselves. So here we go. It's another one. You can actually see the difference in refractive index here with a difference of color. I'm going to go to bright field. So that's bright field now. That's the way that you should also be able to see it. I mean, I'm using here concentrated salt solution. I mean, this is, yeah. Maybe some of them are already having some problems. And yet another one. It could maybe also be that maybe it's not properly mixed. But you see actually the differences in refractive index. That's why you see the changes in color. Yeah, so. But it could be maybe the mechanism that they use to kind of stabilize with a pH, to make them pH tolerant. Maybe a similar mechanism is used also to make them more salt tolerant. I don't know. Yeah. Huh. Honestly, this is also quite fascinating because this is yeah, extremely salty. That'll be, you know what I'm going to do? I'm going to do something very extreme. I'm going to add some more and I'm going to mix it. Maybe this was also... Yeah, some of them start to slow down a little bit. But I have to tell you, I'm fascinated how resistant they actually are. Which causes me to worry. You wonder why? Because this actually means that if they are so pH tolerant and also salt tolerant, what about food preservation? You know what I'm thinking of? I mean, if they're able to survive those extreme conditions as well, yeah. Yeah. Are these the same ones in plant roots? Not necessarily. This was the question here. I suppose you're talking about those nematode worms. There's so many of them, and those nematode worms actually can be found on decomposing fruits. So when they're fruits, you see some of the when they're decomposing fruits, for example, then some bacteria will actually form alcohol as it starts to ferment. And then other bacteria will convert this also into acid and into vinegar, and this is where those vinegar eels also start to live on those decomposing fruits. Yeah, but I think you see already that they start to not look quite as healthy anymore. But honestly, pretty, how do you say, pretty tough guys, I would say. Yeah. So that's a pretty tough guy. You know, what I've done is I added so much salt in the microwave, the salt solution, to dissolve everything. Okay, so yeah. Okay, you see now slowly they start acting a little. Some of them at least start acting a little bit weird. Yeah, and some of them start now shrinking a little bit as well as they lose water. But still, it is surprisingly resistant. Yeah. Thank you. Here are the questions. Thank you. I got again your questions here. Unbelievable. Actually, I wanted to. It's already an hour. I'm gonna move on. I wanna, because I've got other worms as well. Okay. So I also want to show them to you. Yeah. So yeah, you see, what have I learned? What have we learned? Those nematodes are surprisingly, at least those type here, are surprisingly, how do you say, salt tolerant. Wow. Yeah. Now they act a little bit strange as well. You kind of just quill up. Yeah. So next one, my most favorite, everyone's favorite worm, are of course Aeolosoma worms, because they kind of look cute. And I've got a jar here, and there's still several of them here. I have to be lucky a little bit in finding those. These are much larger worms. Yep, now you actually see how they start to shrink up a little bit as well. Some of them actually are starting to become thinner right now. Yeah, yeah. So, yep. Okay, here they go. So let's go back to the desk, and now I need to somehow grab a few of those worms here. I need a, how do you call this? A pipette. And let's try to. This is now a question of luck, because the concentration is not quite as high. So I'm going to. I hope that I'm able to get some. To see. Yeah, I think I have a few here, one here at least. Okay, I'm going to see if I'm able to find something here. Let's remove this here. And the scope view. Oh yeah. Now it's a question of luck if I'm able to find one. Ah, here's one. Why is this? I need to. Here's one in a drop of water. Isn't this nice? Okay, yeah, this guy's caught in a droplet of water. These are the so-called segmented worms. It belongs to the oligochaete worms. It's a segmented worm, so you see it has these tiny bristles here as well. Let's do it like this. Here, let's add a little color. Here we go. Yeah, and you can see. Well, I've been feeding it just yesterday. So I added a whole bunch of yeast. So you see the inside of the intestine is quite dark, probably with the yeast. Yeah. This one, you see the bristles here, right? So it doesn't have legs as such, right? Yeah, and it's also known as the oil droplet worm. And here it's moving around fairly quickly, again without a cover glass. Okay, so what I'm gonna be doing now is again low magnification like this, and we flip out the, here we go. And let's see now what happens, how salt tolerant those guys are. And I have this feeling that maybe they're not so salt tolerant. Yeah, so let's try it again. wipe this here. The pipette is a little bit dirty now. So I need a, what do you need? I need a pipette tip again, and let's add again a little bit of concentrated salt. I'll be adding a little bit now, and let's see what it does. How salt tolerant are those guys here? Okay, yep. Not so salt tolerant. Okay, I think you get the idea. This was a quick end. Yeah. That's it. Ah. This is it? Okay. Yeah, that's kind of shows that it didn't like salt. Huh? So why are the worms so different? Well, this tells us a little bit something about the environment that they're living in, because these, of course, are freshwater worms. They're not exposed, obviously, to high salt concentrations. While the vinegar eels, they have to be exposed to alcohol because of fermentation, fermenting fruits. They have to be exposed to acid when the alcohol is converted, oxidized to an acid. Right, so they have to be used to these extremer conditions. But those freshwater worms, they don't know salt, and that's why there is no need to be able to deal with it. Yeah. So this kind of shows a little bit how those organisms are adapted to their environment, whether they're able to withstand something. It's a little bit maybe similar to the cheek cells. The cheek cells, probably because they're always exposed to different types of environments. Sometimes we eat salty food and then we drink water again. So a very large different salt concentration. So the cheek cells have to be adapted to that, right? Because that's the environment of these cells. Yeah. So that is, I think that's the short one. So maybe the next time I probably should not use such a high salt concentration right from the beginning. Yeah? So, yeah, a murder under the microscope. You're not gonna like the next one. Because I've got others here. Daphnia. And if you feel sorry for those guys, then let me tell you, they would be eaten by fish anyway. So you see, they're gonna die anyway. Well, I don't have fish, but that's kind of the thing. I have no ethical problems with that, because these little critters don't have a consciousness. They don't have fully developed brains. Yeah, they're not conscious in the way that we are, and therefore, I have no problem with those. Okay, let's see if I'm able to get dupe. Here we go. Okay, here's one. And the population sometimes collapses anyway, and then hundreds of them are dead, and then all of a sudden you have more of them. Yeah. So Oh, you didn't see that because I was. But look what we've got here. Maybe we're able to see it better now. Ooh, look at this. Of course. Here it is, Daphnia. No, it's yeah. Oh yeah. Very nice. Can we do some dark field? No, not so good. That's better. This actually looks nice now. Okay. Let's focus. Yeah, the problem is the dynamic range is a little bit of a problem because now it's overexposed. Yeah. So. Is it hatching? Nope, because I tell you why. I do not see any neither eggs nor do I see any babies, and they're usually in the so-called in the brood pouch, which is empty now. Because here in the back, usually this is when where you've got the offspring. Yeah, and then some of them actually give live birth even. Also made a video on that. But yeah, so what I'm going to do now is I'm going to use a very low salt concentration to 12.5%, because I think that they might not like too much salt. It looks, they are related to the shrimp. Okay, so these are water crustaceans. Yeah, so in that sense, if you say it looks like a prawn, like a shrimp or something, you're not wrong, because they are actually related. Yeah, to those. Yeah. What is this over here? Do I have another one over here? What is this? No idea. This seems to be some kind of weird dead matter. Okay. Again, no cover glass now. Okay, and let's do it like this, like this. Let's add a little color. Yeah, that's because of the setup that I have. So the color is of course not natural. But I'm going to now use my 12.5% of salt. So this one is much more dilute. But there's a problem with the pipette tip. It doesn't suck up anything. Let's try another one. Here we go. This one works better. So 12.5%. Let's see how salt tolerant this one is. So this is not concentrated salt solution. This is much less. Okay. Because yeah, it might take a little bit of time. You can do all sorts of weird experiments with those guys. You can add caffeine, you can add alcohol, you can add all sorts of different substances and test how actually they respond, because they do have a simple nervous system, and therefore those substances like caffeine and also alcohol, they do have an impact on those. I'm reading right now. They cannot make a affordable and inexpensive differential interference contrast, DIC. I agree. And I also do not see a reason why this is not possible. There, I can only tell you can imitate DIC using so-called oblique illumination. And I don't know. I think this might be a marketing thing. I know that the company Euromex has DIC microscopes also available. And then, of course, the big companies like Olympus, Leica, Zeiss, Nikon, they have also DIC setups, but this is pretty expensive. Yeah? See, the 12.5% seems to be okay. Yeah, that's interesting. So what I'm going to probably be doing is I want to go up a little bit with the concentration. I've got now the 50%. Salt. So let's see if this. So they seem to be quite a bit. I don't know. Up. You see again the differences in refractive index of yeah. And I can imagine maybe that it depends also a little bit on how quickly they're getting used to the salt, because of course if they're able to change also their physiology around, but this might take a little bit of time. And if you see all of those lines on the outside, that's basically the salt water mixing in. Yeah, I tried oblique illumination. I didn't like it much. It's oblique illumination needs to be adjusted. I agree. Yeah, but I have seen on a German microscopy forum some folks who've really gotten very nice results. Yeah. So, interesting. So, what do we learn here? I mean, at least. So, you know what I'm going to do? I'm going to go now. Now I'm going to go concentrated salt. Okay. So you see that. Maybe they're also getting a little used to, yeah. Yeah. No, now you see that the movement, well, I don't know. It might take, especially for these slightly larger animals compared to the others, might take maybe a little bit more time until actually they're able to, until the salt reaches all of the parts. But you see, yeah. Still moving, still going. Yeah. It got more dense from the inside. It could be actually that maybe it's kind of contracting a little bit on the inside to prevent exposure to the salty environment. Yeah. And see the antenna also not stretched away so much anymore. Yeah. I think the bottom line is that salt balance, osmotic balance is pretty important for living things, obviously. Yeah. This one over here on the back, this one over here is actually the heart. Yeah. And there's taking in the food over here, and that's the intestine. Yeah. Yeah. So I don't know if I'm able to save it again. So I'm gonna add a little bit more water. I'm gonna take away the other salt. I'm gonna add some more water. Okay. Maybe I'm able to... Or maybe it was already too much for it. Yeah, we're gonna see. Yeah. So let me check the time. Yeah, what I'm gonna do now is, as we watch this little critter here, yeah, that here, let's put this with this here is the eye. Yes, that's the eye over here. Yeah, intestine in here, the antenna also for moving around. Yeah, and yeah, they're yeah, and what they do is they are able to reproduce very rapidly asexually by parthenogenesis. This means that a female will form eggs in here, and then it will give birth to, I don't know, 6, 7, 8 babies. I've seen this already happening. Yeah. So pipette size that I'm using, this is up to 50 microliters. Yeah, and yeah, so you can adjust the volume here by turning this. Yeah. So what I'm going to do now is the following. I'm going to use the next couple of minutes maybe to actually have a look at some of your comments or questions that you might have had. Okay, let me have a look here. The desk again. Yeah, I noticed gradually that you made a community and keeping it so... Yeah, I usually wake up in the middle of the night to watch. Thank you very much. You should get some sleep, honestly. It's nice, but that you're actually staying awake. But if you basically watch it later, it's also fine. I mean, usually, as a matter of fact, during a live stream, I usually have about 60 people maximum at the same time. But usually, after a couple of days or week, I have around 1,000 people who've actually seen the live stream afterwards. Yeah, because the live streams are available on YouTube. Yeah, and England, as always. Yes, thank you very much. Yes, I got the email. A couple of when I put the sushi under the microscope 2 weeks ago, there were these strange orange dots inside the ginger. Okay, and thank you very much to Ever Small Jesse that because he did some research and basically these are actually structures in the sushi. Never worry about to repeat the same experiments. If you only do an experiment once, you learn nothing. We all know that results can be different depending on many factors. Yeah, thank you very much. It's like this, that ultimately everything has already been sometimes done before. What I'm doing is some kind of variations of it. Yeah. Do you know that the red onion skin extract is used to bio-UV filters for solar? No, I didn't know that. Really? Okay. Interesting. Bio-UV filters. So the red pigment, the so-called anthocyanins. If you lower the condenser in bright field, does the contrast on the cheek cells increase? No, because the contrast increases if you close the condenser diaphragm. Yeah. If you lower it, then sometimes what will happen is that it will become darker. Yeah. So yeah. I have a feeling that the cheek cells are more resistant to salt concentrations. Yes, that's what I think as well, even though I've read that this is apparently not everyone was able to repeat that. Yeah. My microscope's just a cheap one. I chose a trinocular one, and I changed all the objectives from non-standard to plano acro and enhanced slightly the lighting. I only use bright and dark field. Yeah, you can. With many microscope, you can upgrade them because they are basically adhere to a standard. How's the terrarium going? Are the microorganisms still alive? I don't know if they're still alive, but I'm quite sure they are. Look, a few weeks ago, I made this terrarium here, this mini terrarium, and every now and then some insects start to fly out because some insects were. But I'm quite sure that actually there are quite a few microbes now, nematode worms, and actually they're not microbes, they're just microanimals, are in here because it's been now decomposing the wood and the moss is growing. So I wanted to actually put this again under the microscope in an upcoming live stream, to check on how it's doing. Yeah. Yeah, so let's have another one. So which camera is he using now on his microscope? What I'm using right now is a DSLR camera. This is an Olympus D90D camera. And I'm connecting it to this to the software using HDMI. So basically, it's an HDMI stream, and that's how I'm doing that. I'm converting it to USB. Yeah. What active cultures do you have at home? Are you able to keep any of them for a long time? I have a few of them, and what I usually do is the following. I usually keep some pond water samples in a jar for several months, and then if I see that it doesn't turn out nicely, I just throw them out again. So it's always a, how do you say, a process, a continuous process where I exchange them. Yeah. Yeah, after this video, I think I have to search for a microscope at home. Okay. Yeah. Ultimately, because people want to buy microscopes, ultimately, I have to be honest with you, because people sometimes ask me what's the best brand. Honestly, many of them are quite similar, and it doesn't really matter a huge deal which brand you buy. You just don't invest too much money at the beginning. Start small, and as you learn more, you're just going to buy upgrade sooner or later anyway. Yeah. Yeah. So that is and I've got an Olympus BX53. That's the name of my microscope, and and what I have is, in case you just wonder, what I have is I've got I don't know, I've got a camera mounted in a small camera, so that you're able to see the stage here, and then the one over here, that's over the HDMI feed. And now the Daphnia died. Okay, I was not able to save it. Yeah. So tough luck for the Daphnia. Okay, but essentially that's and everything is connected into how do you call it, into OBS Studio, and that's how I'm doing the live stream. Yeah. So I'm just looking if there are any more comments here. Okay. Yep, conversations going on inside the community. Christiansen illumination, yes, which was mentioned over here. I have tried this as well, and indeed it actually does work. Christiansen illumination actually is oblique illumination, where there is a diffuser. There's kind of this diffusing tape, and there's the question which brand. Just any type of tape that it has this white diffusing thing should work. Yeah, and with this diffusing tape, it's surprising. It actually really improves it. Yeah. Sugar as well. There's also the question: what about salt and sugar? Both salt and sugar are osmotically active, and this is, by the way, the reason why. Let me put this other thing away. Why, when you want to preserve food, you either add a lot of salt. Salt concentrations, or a lot of sugar. That's why, for example, jam and so on is able, or honey is able to store well for a long time, is because as soon as some bacteria fall into the jam, then they immediately lose water and they die. Yeah, most of them at least. Yeah, so I go on. If there are any further questions, shrimp, nitric acid, DIC. Yeah, I have to tell you the following. I know I'm going a little bit maybe off topic here as well, because you see, there are lots of microscopes available, even online on Amazon and so on, way more than used to be. What these microscopes, however, all have in common is that they're generally not easily upgradable. Right, so you basically buy a microscope out of the box, ready assembled, and that's pretty much it. And if you want to get spare parts, it's sometimes difficult to get them. Right, I mean, for example, we've in school we've ordered a set of a whole bunch of microscopes a few years ago, and we said we don't need all of the objectives, we don't need all of the eyepieces. Can we get a price reduction a little bit because we want to reuse the others from the existing microscopes? But the problem is we could not get partially assembled microscopes because they all sold them ready-made and packaged right from the company, right? So that is a little bit the problem. DIC and the cost of DIC. That's a little bit unfortunate. And so that is the thing. And I think I'm just going to slowly call it a day again. I mean, I still maybe have 5 minutes or so. Just but I think I think I'm going to call it a day. It's almost 1.5 hours anyway. If you have any recommendations on what you want me to put under the microscope, please feel free, of course, to I take your recommendations. I know that last week I promised that somebody requested that I put some paramecia again under the microscope for galvanotaxis. This basically means in an electrical field. I didn't get the paramecia culture together yet, and I need to get a slide with proper electrodes, so I'm working on that as well. That's basically, we'll see. I'll see if I'm able to manage this by next week or not. Okay, so I am going to call it a day for today. I did not look at the yeast, but that's okay. I'll save it for next time, I guess. Okay, and I think I'm going to indeed call it a day. Hope that you liked the live stream. I will be, just for your information, that's also something I wanted to say. I'm currently making more videos, maybe 1 or 2 videos per week in addition to this live stream on this channel. But those videos will be also a little bit live stream like a little bit. So it will be mostly unscripted, and I will just record something. And I've already finished a video where I put banana, where I'm extracting banana starch grains. And I'll show you how I do that, and I'm going to be uploading that video also in a few days. Okay, so please keep on checking. There will be a banana video coming up in a few days about the starch grains. Yeah, copper tape. Thank you, copper tape. I didn't think of that. Maybe self-adhesive copper tape would do the trick. Yeah, All the best. Bye bye. See you around. Bye bye.